Thank you. So I am going over the syllabus. There's just 2 changes that I had to make, or 3, I think. Yeah. So the first change is the lab that we did last week. I had put 50 points, and this is before I had written the syllabus. So I needed to change it to 25 points. So it's just worth half of what it was before. So it's not worth as much of your grade. So all of our labs are going to be 25 points. And another change was before Dr. Radwan left, his first lab, he had labeled it lab number 3. And I think that was maybe the first activity you guys did. So our lab, I had numbered lab 4. So because I took over and we're basically starting from fresh, really, I changed last week's lab to lab number 1. And so today's going to be lab number 2. So next week is going to be lab number 3. So those are those changes I made. So these are just the labs that I came up with. There's 3 types that we're going to be doing. The first one is Labster Lab, so simulations, and then Labster has quiz questions that they give you in the simulation. So there's no quiz or worksheet when we do a Labster Lab. The second type is I'm going to be going to Simmons twice this semester to record myself doing an experiment, and then I'll post it to YouTube. So for those type of labs, I'll just put a few questions on 2U as a quiz, just so I know that you watched the experiment and understood what was happening and the results. And then the third type of lab we're going to be doing is another video-based lab. It's called Jove Video. So Jove videos give you, it's like usually a 10-minute video. They're pretty short and they kind of go over a topic. Like in this video, it's going to be about TLC, thin layer chromatography. And then because they're so short, we'll do a group worksheet. So in your breakout room, you'll watch this 10-minute video, and then with your group, complete a worksheet together. And then you only submit 1 worksheet per group. So what I would do is when you go into your breakout room, designate someone as your scribe, and then that person will complete whatever you're submitting. It can be a Google Sheet, which I think might be the easiest because then you could share it all together. And then they put all of your names on it, and then they submit it to their Google Drive folder. And then I'll be able to see that, these 5 students completed this and they all get that grade for the group worksheet. Okay, so those are the 3 types of labs we're going to do. There might be some labs where we'll do a short breakout room activity before you start either watching a YouTube video or maybe starting a Labster, and that just depends on what we're talking about that day. So in those cases, it would be like a 10-15 minute activity that you do with your group, and just so you can talk to someone and you're not just watching things. Okay, so that's what I've come up with for the semester. And then I wrote when we are not having labs. So on the 15th, there's no lab. Then on the 15th of April, it's an administrative Monday, but it falls on a Thursday. So we have lab that Monday. So I decided not to do 2 labs that week. I thought it would be a little much. So the next lab after the 12th isn't until the 19th. So on this administrative Monday, on that Thursday, we don't have lab. And then last thing is, instead of a lab exam or a project or anything like that, I'm going to be doing a lab quiz. It's just 25 points. And I'm going to need to make it timed. So I'm leaning towards 45 minutes. And it'll be just 10 to 12 questions, so 25 points. It shouldn't take you that long. And then if you have extended time, just let me know so I can adjust, your time for that. But about 45 minutes, 25 points. I'll be posting a practice quiz on 2U, and then the lab quiz will also be on 2U. So the practice quiz that I'll post on 2U will have similar multiple choice questions to the end of semester lab quiz. So you'll get some practice taking it, and yeah, so hopefully that will be okay for everyone. So that's the schedule. Any questions about the schedule? Okay.
So I just have a quick question. So for today's lab, it's due like next Monday that we meet, and last Monday's lab is due tonight. Is that how you're doing the due dates?
So for Labster, since you'll be doing it in your breakout room and there's no extra work, you could just do it today and have it due. So for
Okay.
the Labster ones, I think we'll do them just today. We'll have them in today. If you have a problem and you can't get it in, I'm very flexible. Like, you can email me and say, hey, couldn't get it done, XYZ. You can turn it in. But like the lab report from last week, there are multiple pieces. That's why I gave that week. And then the group worksheet, since you are working with a group, that's probably something you would want to post the day of the lab, since you're doing it with other people in everyone's schedules. Yeah, and then the 2U quizzes, when we do a YouTube, those are short, so those would be like due the day of as well, because a lot of people can't do work outside of our lab time. Does that sound okay?
Yeah, I was just making sure because it's, yeah. All right, thank you.
Yeah. So yeah, the Labster stuff's flexible. Quizzes, we'll do a day of. Group activities, day of. And then if we ever had to do a lab report again, like, I'll give you a week for that. But I don't foresee doing another lab report, because that was a little bit more in depth than I would like. Okay, so yeah, that's a good question. So any other questions about the schedule there? Okay, awesome. So that's just my info there. And Angela, who is our awesome TA tonight. And then this is the lab format. So I just kind of described the 3 types of labs that we're going to be doing. Attendance. So again, I'm flexible. So if you're going to miss a lab, just email me. You could either come to another lab session. There's a lab session at 9 and 11, or I record this 6 p.m. So people who missed earlier today, this is what I send to them. So if you miss a lab, I'll send you this recording. You just have to email me, because I need to know to send it to you. I'm not posting them on 2U. Okay. And then there's just the info about the lab quiz. And then you can drop the lowest lab grade out of the 10. So you are only counting 9 out of 10 labs, and it's 225 points. And then it's 25 points for that lab quiz. So it's 250 points, and Professor Levy, it's 25% of your grade is what he calculated. So 250 points is the lab. Okay. Any other questions about the syllabus before we talk about the experiment or Labster? Okay. So that's on 2U if you don't already have it. And I will bring up the lab 2. Okay. So this is also on 2U. I wanted to give an overview of the Labster in case this isn't your first time, and give you some background. So in today's lab, we're looking at chemical tests for functional groups. So the functional groups we're interested in are alcohol, phenol, and carboxylic acid. So you haven't really gone into depth about carboxylic acids in lecture yet, but the purpose of the lab is just to talk about chemical tests. So what you would do in a lab to test for a functional group, not necessarily knowing anything about carboxylic acids or alcohols or phenols. So these are just the objectives of the experiment. So what you're going to be doing is you meet someone called Simon, and they are given a painkiller for a toothache, and they were told that it has salicylic acid. It's salicylic acid, which is what is in aspirin. And we're actually going to be doing an aspirin. I'm going to be demonstrating an aspirin lab later in the semester, but aspirin is a pain reliever. So they're just not sure if what they were given contains salicylic acid. So you'll perform 2 tests on the unknown compound to see if it contains salicylic acid, and you'll do that by running tests about their functional groups. So just a little explanation about chemical tests for functional groups. They identify unknown compounds. So if you're doing a chemical test for an alcohol, for example, if your unknown contains an alcohol group, then it'll give you a positive result. So here's just an example that's going to be in the lab, alcohols. So they have those hydroxyl groups in them, which is the OH. 2 examples, ethylene glycol and xylitol. So ethylene glycol is an antifreeze. If you've ever watched a murder documentary, that's one that people have used to poison people slowly. Yeah, morbid, but. And then xylitol is in like gum and toothpaste, and it's really good for your teeth, but it's really toxic to dogs. So those are just those hydroxyl groups on there. So if we were doing a test to find out if we have an unknown substance, we think it's xylitol, let's test xylitol and the unknown substance. And if we get a positive result, we could do some more tests, but maybe we'll be able to figure out if it is xylitol, if it's testing positive for hydroxyl groups. Okay. Another example, alkyl halides. So you're going over those in lecture. I think you went over them today in lecture. So it's not part of the Labster simulation, but I just wanted to give a little bit of background into a test you would do for alkyl halides, because you are learning about it in lecture. So if you remember what you're talking about with Professor Levy, those are compounds that have a halogen in them, and it takes place of one or more hydrogens in the hydrocarbon. So halogens, fluorine, chlorine, bromine, or iodine. So an example of those is CFCs or chlorofluorocarbons, like refrigerants or aerosol sprays or air conditioners. So there's an example of a alkyl halide. Another one is chloroform, which is interesting because it has a history in medicine. If you've ever watched like an old movie where someone's kidnapped and they put like a towel soaked in something against their mouth, it might have been chloroform, and it like knocks them out. So it used to be used in surgery and for pain as an analgesic or anesthetic, and it stopped being used in the 20th century because it was showing a carcinogenic effect in mice and rats. So that's chloroform there. So one example of a chemical test for alkyl halides is something called a silver nitrate test, and this is what results would look like from a silver nitrate test. You basically add nitric acid and silver nitrate to your test tubes, and then it will turn these colors if those alkyl halides are present. So if chloride's present, it'll form that white precipitate, bromide, cream, and then iodine, that yellow, if those alkyl halides are present. So it's not in the Labster, but that's just some info about alkyl halides and a chemical test for them. All right, so into the labs, we're almost done with this intro part. So the first test you're gonna do is a bromine test. And this is kind of Labster just wants you to practice running a chemical test. It's not helping you with helping Simon yet, it's just giving you practice. So basically the bromine test is testing for unsaturated hydrocarbons. So the bromine water is this orange color here, and then it becomes clear or it loses its color when it's in the presence of an alkene. So an alkene has that double bond. So you're going to see that one of your test tubes loses its orange color, and that's how you determine what's in your test tube. And then once you do the bromine test, you're going to do the 2 tests on your unknown to test if it's salicylic acid. So the first test you're going to do is called a steric ammonium nitrate test, and that's testing for alcohols and phenols. So the phenols is right here. So it's a hydroxyl group that's attached to an aromatic hydrocarbon ring. And they're mildly acidic. They can cause chemical burns. And I talked about this in the earlier labs. I don't know if anyone's interested, but this is I was really into skincare in Chem 111, and that was one of the projects that students presented. So I've heard a lot about this product that celebrities and facialists use. I don't know if anyone's heard of it, but basically it's like a favorite. It's expensive. It's like over $100 for a bottle, but basically it was developed, I think, in like the 50s, 60s, sometime around there. And it's the one that's the 1970. They actually put phenols in it. So there's some controversy about having phenols in products that you're applying to your skin because it can cause chemical burns. And the purpose of this product is to, I think it's like a peel, like a chemical peel. It's supposed to like rejuvenate your skin cells and like take off a layer, something like that. I don't know the background of it, but people seem to love it. So they took out that phenol and made this new product without it that apparently works the same way. I've read that it smells like spoiled milk, which doesn't surprise me because there's lactic acid in there. There's onion bulb. There's salicylic acid in there too, which is what we're talking about today. So that's just an interesting thing that I remember reading about phenols in skincare. And then I'm going to the lab tomorrow to record a video. I'm going to look to see where we keep our phenol, if there's any at Simmons, and what it says on the bottle, because I'd be interested. So anyway, fun fact. And then the steric ammonium nitrate test is going to form a dark red color when it is added to an organic compound that has an alcohol or a phenol. So you'll see that color change happening in your ammonium nitrate test. So once you're finished with that test, you're going to do the last one of the Labster. It's the sodium bicarbonate test, and that's used to determine if carboxylic acids are present. So just to be aware, since you haven't covered in lecture, there's a question about carboxylic acids and what a COOH group is. So this COOH group is a carboxylic acid. That's how you would identify a carboxylic acid, by this group here. So if you look down here at acetic acid, which is vinegar, you'll see the COOH. That's the carboxylic acid there. Ascorbic acid also has a carboxylic acid group in it. Where are we? Here it is right here. So there's our C double bonded to an O, and then there's our OH group there. Right? Is that right? Because there's C double bonded to an O. Maybe that's where it is. I don't know. This is a better picture. I don't really know where that would be a COOH. Anyway, so just know that the COOH group is a carboxylic acid. So if in the sodium bicarbonate test, you're going to see bubbles forming. So when those bubbles form, you know that there is a carboxylic acid present in the compound. So here is the salicylic acid. So that is what you're testing for. Yeah, this is a carboxylic acid here. See the C double bonded to an O and then to an OH. So the OH is attached to the C, and then the C is attached to a double bonded O. So if you notice the phenol group here, hydroxyl group there, the alcohol, and then the carboxylic acid right there. So knowing the structure and those functional groups that are present on it, you should be able to hypothesize what your test results are going to be based on that other information I gave. Okay, and then once you're finished with the simulation, you're going to be in the breakout rooms. Once you're finished, you're all set. You don't have to come back in, or there's nothing to complete, and it's 25 points. So I'm going to split us into breakout rooms,